All right, welcome to part 4 of Mod 11 on cell division. Sorry that this lecture ended up being 4 videos, but I'm trying not to go too long on the individual videos. This last one should be a little bit shorter. In part 4, we're going to be talking about alterations of chromosome number and structure. Accidents during meiosis can alter chromosome number. An abnormal chromosome count is the result of non-disjunction, which can result from either the failure of a pair of homologous chromosomes to separate during meiosis 1. As you can see right here, meiosis 1, this pair of homologous chromosomes failed to separate. This one on the right should have moved to the right of the cell, but it did not. Non-disjunction can also result from the failure of sister chromatids to separate during meiosis 2. So here we have meiosis 1 occurring normally, but if the sister chromatids fail to separate, they should each go to one pole of the cell, but over here these sister chromatids did not separate. That can also result in a non-disjunction. We're going to walk through the non-disjunction in meiosis 1 first. So if the non-disjunction occurs in meiosis 1 and the homologous chromosomes fail to separate, in meiosis 2, we'll end up with one cell that has 3 pairs of sister chromatids and then one cell that has 1 pair of sister chromatids. But other than that, meiosis 2 will proceed like normal. As a result from that, all of the gametes produced as a result of non-disjunction in meiosis 1 will have abnormal numbers of chromosomes. These ones each have 3 chromosomes, so that's n plus 1. They have an extra chromosome, and then these 2 chromosomes over here only have 1 chromosome, so that's n minus 1. All of these are abnormal gametes with an abnormal number of chromosomes. So if non-disjunction occurs during meiosis 1, all of the gametes produced from that meiosis will be abnormal. However, if non-disjunction occurs during meiosis 2, so meiosis 1 occurs normally, and then in meiosis 2, we have this non-disjunction of the sister chromatids, then only half of the gametes will be abnormal. These 2 over here will have 1 gamete with 3 chromosomes, so n plus 1, 1 gamete with 1 chromosome, so n minus 1, and then over on the right, all of those gametes will be normal. So half the gametes will be normal with n amount of chromosomes, and then half the gametes will be abnormal with either n plus 1 or n minus 1. In 10 to 30% of conceptions, I believe, non-disjunction does occur, and the primary reason for miscarriages is non-disjunction occurred during meiosis, and the zygote or the developing embryo has an incorrect number of chromosomes. We'll talk a little bit later in this lecture about some survivable genetic conditions where individuals will have different than the normal 46 chromosomes, but for the most part, having either 1 fewer chromosome or 1 more chromosome is fatal and results in a miscarriage. To look at an individual's chromosomes and see if they have the normal amount or fewer or more, we use something called a karyotype. A karyotype is a photographic inventory of an individual's chromosomes. To prepare a karyotype, it's easiest to do using white blood cells. In the process, white blood cells are isolated, stimulated to grow, arrested at metaphase, and then photographed under a microscope. This image here just kind of walks through that process. So we have a blood culture. That blood culture is then put into a centrifuge with fluid. It's spun around. We end up with packed red and white blood cells down here at the bottom of the tube. A hypotonic solution is then added to the mixture, and then the white blood cells are isolated. A fixative is added, and then those white blood cells are added to a stain, which is then photographed under a microscope. This is an image of someone looking at those white blood cells under a microscope or the stain under a microscope. The chromosomes are arranged into ordered pairs in a karyotype so that any chromosomal abnormalities can be detected. This is what a karyotype looks over here. Notice that the chromosomes are stained to see the different banding patterns. If we zoom in on the homologous pair 1 here, we'll see, it's kind of hard to see in this image. This is the homologous pair, which is what you see over here on the right. The sister chromatids are a little harder to see over on the right, but if you zoom in on the left, you can see that the centromere is right here and there are 2 sister chromatids. Again, centromere is here, 2 sister chromatids. This overall is a pair of homologous chromosomes. In the karyotype on the right, it's harder to see the sister chromatids, but each homologous pair has 2 sister chromatids here and then 2 sister chromatids here. This karyotype has all the homologous pairs numbered from 1 through 22, and then the homologous pair 23 is the sex chromosomes. We talked about this in the last video. An individual who has the sex chromosomes XY is male at birth, and then an individual with 2 X chromosomes is female at birth. So we can see at this one, the X and the Y are not technically homologous. The X chromosome is a lot larger than the Y, but for the purposes of meiosis, they do behave like homologous chromosomes. But this is a male because we have the XY chromosomes here. A checkpoint question here. How would the karyotype of a human female differ from the male karyotype in figure 8.19? So we have a male because it has an X and a Y. How would a female karyotype differ from that? A female should have 2 X chromosomes and no Y chromosome. Instead of an XY combination for the sex chromosomes, there would be a homologous pair of X chromosomes. So 2 XX, 2 of those longer X chromosomes. Karyotypes allow us to look at an individual's chromosomes, and they can be used to diagnose certain disorders or to show certain disorders. One of the most common examples of an extra copy of a chromosome is trisomy 21. Trisomy 21 is when there's an extra copy of chromosome 21, and this causes Down syndrome. So trisomy 21 is the most common chromosome number abnormality, and it results in a condition called Down syndrome. It results when there are 3 copies of chromosome 21. You can see right here, instead of the usual 2, there's a 3rd copy. Trisomy 21 is one of the only examples where you can have an extra chromosome that's not an extra sex chromosome and it not be fatal. So trisomy 21 is one of the only chromosome number abnormalities that is not fatal. An embryo will survive to full term and will be born even if they have 3 of chromosome 21. There are the characteristic symptoms associated with Down syndrome, including the flattened kind of nasal fold, and then there's folds inside the eyes. Another interesting thing about Down syndrome, this graph is from a study where they were looking at the age of the mother and then the number of infants with Down syndrome per 1,000 births. You can see that as the age of the mother increases, the occurrence of Down syndrome also kind of exponentially increases. So the age of mother, below about 35, we see a pretty low risk. It does increase slightly as you're getting up to 35, but very minimal. But then from 35 to 40, we see at 35, it looks like it's about 3 or 4%. And at 40, we're already at, sorry, it is not percentages. It is number of infants born. So at 35, it looks like there's about 3 infants per 1,000 births born with Down syndrome. Whereas at age 40, that jumps to 10 infants with Down syndrome per 1,000 births. And then at 45, it's about 31 infants with Down syndrome born per 1,000 births. So the age of the mother does correlate with Down syndrome occurrence. Abnormal numbers of sex chromosomes do not usually affect survival, unlike abnormal numbers of the autosomes. Like I just mentioned, Down syndrome is one of the only syndromes where you can have an abnormal number of the autosomes and still survive. The sex chromosomes, you are able to have either more or less sex chromosomes, but they do result in syndromes. Non-disjunction of the sex chromosomes during meiosis can result in individuals with a missing or an extra X or Y chromosome. In some cases, such as XXY, this leads to syndromes that can affect the health of the individual. In other cases, such as XXX syndrome or XXX, the body is completely normal. On the next page, I kind of have a summary of these different syndromes associated with either missing or extra sex chromosomes. So remember, a normal female is XX and a normal male is XY. The first syndrome is Klinefelter syndrome. Based on genetically, the individual is male, but the sex chromosomes are XXY. The origin of the non-disjunction is going to be meiosis in egg or sperm formation because there are two X chromosomes here. There's an extra X. The symptoms, sterile, underdeveloped testes, and secondary female characteristics. The next sex chromosome abnormality is XYY. So in this case, there's an extra Y chromosome that has to come from meiosis in sperm formation in order to get that extra Y, because a female XX would not be able to pass on a Y. There's no syndrome associated with this. The individual would be a normal male, and there are no symptoms. Another sex chromosome combination, we can have XXX or XXX. There are no syndromes associated with this. An individual would be a normal female. The origin of this can be meiosis in egg or sperm formation. The only symptoms is someone with 3 X chromosomes might be slightly taller than average. And then finally, we have Turner syndrome, which is when an individual only has 1 X chromosome and then the other sex chromosome is missing. This is the only known instance where having 1 fewer chromosome is not fatal in humans. So an individual with Turner syndrome would only have 45 chromosomes. Only time that's not fatal that we know of, to only have 45. The origin of non-disjunction, again, can be meiosis in the egg or the sperm formation. The symptoms of Turner syndrome are the individual is often sterile and they have immature sex organs. So this kind of wraps up the abnormalities of chromosome numbers and talking about karyotypes, and it wraps up our whole discussion on cell division. So I will see you guys for the next video. Module 12 is a lecture exam week, so I'll see you in Module 13.